Welcome to lesson 6.9. This lesson is about soil protist ecology. So we'll look at how protozoa or soil protists function in the soil ecosystem. We'll look at their feeding preferences and their vital role in nutrient cycling, how the function of soil protists by feeding on various organisms such as fungi and bacteria really influences and the impact that has on nutrient cycling to the plant and to the entire soil as well as above ground ecosystems. Okay, so this is lesson 6.9. Let's get to it. So let's first look at how soil protozoa or soil protists interact with the soil matrix that they find themselves in and in which they function. The soil protists are more abundant in the rhizosphere compared to the bulk soil, which makes sense because most of the bacteria hang around the roots. They are a significant component of soil biomass, bioactivity in soil, and soil biodiversity. They're heavily dependent on soil moisture, texture, and structure, and they are of a small size, so they can access small pores in the soil matrix, if these pores are available, of course. Naked amoebae, such as the one pictured on the right, are shapeshifters, so they can actually squeeze into tiny pore spaces. Soil ciliates are often much larger than the other soil protists, and in soil they take on an elongated and narrower shape versus when they live in aquatic environments. Flagellates and naked amoebae in agricultural soils can reach up to 100,000 cells per gram of soil, and flagellates and amoebae dominate cultivated clay soils. Flagellates can be found in drier soils versus ciliates, which require wetter soils, and are also able to withstand reduced oxygen conditions that wetter soils provide. Larger flagellates, testate amoebae, and ciliates are found in coarse, textured soils. And of course, it is very important to have soil protozoa, soil protists in soil ecosystem, as they are a very important food source for nematodes and rotifers, which are multicellular soil-dwelling organisms. Soil protists, like other soil organisms, depend on the abiotic conditions in the soil. So, for example, the level of oxygen and water and the balance between those two is very important to the survival and proliferation of soil protists. They are aquatic organisms by nature, so they live within pores and between soil aggregates. They can be found in temporary anaerobic microsites in well-aerated soils. To tolerate anaerobic conditions in the soil, say the soil is waterlogged and oxygen has been used up, they can encyst, so become cysts. And some of them don't mind microaerophilic conditions. So when soil ciliates start to appear in soils, that's an indicator that the soil oxygen conditions are reduced. And it's often very important to bring oxygen back up in order for the other protists to be able to survive, as they tolerate less anaerobic conditions or only microaerophilic conditions rather than anaerobic conditions. There are also pore size restrictions. So the smaller the pores, the larger protists won't be able to get in. And that, of course, relates to soil structure, what sort of size the mineral component takes on, for example, clay versus sand, but also how the soil is structured in terms of aggregate formation in the soil. If there are no aggregates and the soil is just compacted, of course, that pore size is reduced, and therefore certain larger sized protists will not be able to survive. But the soil pore network architecture, which depends on soil texture, structure, and moisture, are very important to the proliferation of soil protists. And they engage in interaggregate travel, which is all moisture dependent. If there's a water column within the channels between aggregates, these protists can move about quite well and therefore function and reproduce across the soil matrix. Their preferred pH ranges from 4.5, which is quite acidic, to 9.5, which is quite alkaline, and that's a huge pH range to exist within. However, 5 years of repeated sulfur fertilizer, which is quite acidic, at the rate of 22 and 44 kilograms per hectare, has been found to significantly reduce pH of the soil from 5.7 to 5.2 and 4.7 respectively, based on these two amounts. And this significantly reduced bacterivorous protozoa, mycophagous amoebae, and microbial and fungal biomass. So both bacteria and fungi biomass as well as biodiversity were reduced as a result of this decrease in pH based on sulfur fertilization. There are also temperature limits for protozoa in the soil, with 30 degrees Celsius being the maximum temperature they can withstand, and they experience death at 35 to 40 degrees Celsius. Some protozoa have very specific temperature requirements. For example, in the Antarctic fell fields, Heterometa globosa has low optimum temperature, and its growth increases, and so does encystment at reduced temperatures. So it actually prefers cooler temperatures, and it just shows that protists, like every other organism, are adapted to specific conditions within which they thrive. And of course, as soil temperature might increase with climate change, these organisms might no longer be able to withstand those temperatures and will either adapt or vanish. So it's very important that to maintain the biodiversity of soil protists, like any other organism, we actually try to maintain, at least to some level, the conditions within which they thrive. And I just want to draw your attention to the image on the left, which is that of different amoebae, and it comes from this website that I got led to via a research paper I was looking at, and it is by Joseph Leidy, drawn in 1879. Apparently this man was an exceptional naturalist, a geologist in the United States, as well as a fantastic microscopist, and has really pioneered early microscopy by documenting and drawing really incredible artwork or scientific images, really, of these incredible creatures that he's found, majority of which are aquatic protists. So I just thought it'd be lovely to use one of his images, and you can look him up further under this link here under the image. So let's look at the soil protozoa or heterotrophic soil protists' functions in the soil ecosystem. They greatly benefit rhizosphere ecological functions because they impact soil and above ground organism communities. Particularly through their cycling of nutrients from bacteria, they feed higher trophic levels. So they are that intermediate between the primary gainers of nutrients, which are the bacteria and fungi that can gain those nutrients from the mineral component of the soil, to then being able to unlock those minerals from those organisms and to release those nutrients for all the other organisms, because they basically, by feeding on bacteria and fungi, they actually crack open the package of minerals that these organisms are. So they mineralize and release plant available nutrients by feeding on bacteria and fungi. They therefore influence soil organic carbon accumulation and stabilization. They are even capable of changing root morphology, physiology, and nutrient absorption of the plant, particularly through their hormonal effect on plants. And they greatly influence soil microbial diversity by preferential feeding on the different microbes or the different bacteria in particular in the soil, and this stabilizes ecological functions in the ecosystem. They connect the plant community and the multi-trophic level cascades. The cascade is basically when predators affect other trophic levels as a result of the organisms they feed on. So in this case, protozoa grazing on bacteria, they will affect the community of that bacteria in the soil and therefore have an effect on the plant community as a result. They suppress diseases, for example, mycophagous amoebae feeding on plant pathogenic fungi, and also have capability of bioremediating or cleaning up contaminated soils. And we'll talk about all of these components in more detail as we move on. I'd like to draw your attention here also to these beautiful ciliate images by the Canadian nature photographer that I have already mentioned, and I'm just using his images because they're absolutely gorgeous, and these are probably once again aquatic ciliates but very worth admiring the beauty of, and many ciliates of equal beauty actually grace soil with their presence as well. So before we talk about nutrient cycling, how nutrients enter soil through soil protists feeding on bacteria and fungi, and particularly on bacteria, let's examine their feeding preferences. And this is in particular related to amoebae and ciliates. So there are many different species of bacteria and protozoa. Amoebae prefer to feed on bacteria and flagellates, but different species of soil protists or protozoa eat different species of bacteria. And experiments have been done to show that 3 different protozoa types fed different species of bacteria actually preferred one species, while some ciliates eat all bacteria. So it just really depends on the soil ecosystem and the feeding preferences of these organisms, as well as on the food available. And of course, soil protists are a fantastic food source for nematodes, rotifers, and microarthropods. So it really just goes to show that everyone's got their own favorite food, and that food type depends on the types of protozoa or protists present in the soil, as well as the foods that they prefer to consume. And of course, this really relies upon the type of plant that is present that feeds the bacteria upon which the protozoa feed on. So you can see how the ecosystem loops back on itself, how each organism present depends on other organisms that are in the system. So let's look at plant available nutrient cycling, or nutrient cycling in general. How do protozoa, and in what way do they deliver nutrients from bacteria, particularly in fungi, to the other organisms, including plants, and of course us as we consume plants? We've already covered the mathematics of how much nitrogen a flagellate can actually access, and that was a few lessons ago when we first began talking about protozoa, so we won't go into that now, but this is the general scenario of how all this occurs. So first of all, there's microbial predation, and nutrients are cycled from the bacteria to higher trophic levels. So that means whatever eats the protozoa that now has these nutrients from the bacteria, those nutrients are now automatically available as protozoa is being consumed. But also as protozoa consumes bacteria, it actually mineralizes those nutrients. So from the organic bacterial bound form to the mineral form. And this is particularly true for nitrogen and phosphorus, which are originally fixed by the microbes. And then as the protozoa defecate those excess nutrients, these nutrients become immediately released for plant consumption, and other organisms can also take advantage of those nutrients, such as bacteria and fungi, for example. They can take those nutrients in that are released as well. So protozoa mineralize nitrogen and excrete ammonium, and this is a source for microbial growth and plant uptake. So that's just to summarize the nitrogen cycle part of it. So mineralization influences the microbe type, so which type of bacteria take up the nutrients, and the fauna type that can feed on those microbes, so the type of protozoa that can actually proliferate. It also determines then the grazing intensity on those microbes and the microbial metabolism in the soil, and also as a result, it influences the soil physiochemical characteristics. So you can see that loop is generated where originally the protozoa mineralize those nutrients, then that mineralization influences the actual soil ecosystem further. So it's just a continuous loop as nutrients become available and influence all the biological and physiochemical characteristics of the soil. So soil protists provide constant excretion of these nutrients and therefore provide active organic matter and enzymes being available in the soil ecosystem. They also control nutrient release and uptake by plants, driving plant community characteristics, and we'll go into this further soon. Additionally, microfauna or soil protists very much influence carbon respiration in the soil and therefore carbon sequestration. So the interesting thing is that soil protists and plants and other soil organisms produce this rhizosphere priming effect. And what this is the acceleration or retardation of soil organic matter decomposition by living roots and associated rhizosphere organisms. So the rhizosphere can increase soil organic matter decomposition by 3 to 5 times, or 380%, or decrease it by 50%, as far as this has been looked at. The microfauna, so soil protists and other predatory organisms, can actually increase the rhizosphere priming effect by 50%, increase microorganism turnover rate, nitrogen distribution between roots and microbes, and the substrate choice and usage, what they actually feed on. And in turn, this really affects microbial activities. So the way that protozoa selectively graze on microbes determines the microbial community structure, and this in turn influences soil organic carbon accumulation and stabilization versus destabilization. So really it just depends on what the plant and microbial ecosystem is like in the soil will influence the organic matter deposition and whether that organic matter stays in the soil as soil stable carbon or whether it is released as carbon dioxide. So it is very important to understand the dynamics between soil organisms, particularly the grazers of microbes such as protozoa, and their influence on soil organic matter and soil organic carbon via their effect on an interaction with the microbial biomass within the soil. And that is largely related also to how they make mineral nitrogen available to stimulate microbial and plant growth, or if there's not enough protozoa, that mineral nitrogen might be missing or might not be sufficient, and this will continually have an effect on the carbon and nitrogen relationship, and therefore on what carbon is sequestered into the soil versus what carbon amount is actually released as carbon dioxide. So these are important questions to ask and study these phenomena in order to better understand how these organisms, along with plants and other soil organisms, can actually influence carbon dynamics, soil carbon sequestration, and carbon dioxide release from the soil. We have touched upon photosynthetic protists on and off throughout this unit. However, it's so essential to stress out just how important these organisms are in nutrient cycling and soil fertility. Because they are primary producers of carbon, being photosynthetic, they directly deliver carbon to soil, and as a result feed a lot of organisms just via their own photosynthetic production of carbon. And this has a flow-on effect that really provides a lot of benefits to the soil ecosystem and other organisms. However, photosynthetic protists are rarely mentioned in soil food web models, and therefore it is very important to mention the actual benefits that these organisms provide to the soil ecosystem. Whether it is microalgae themselves individually living in soils, or whether it is through the algae being engulfed and having their effect in another organism and living in that symbiotic relationship inside of another protist. So, first of all, the fact that microalgae produce oxygen, they are photosynthetic, so they deliver that oxygen to that topsoil layer that they dwell in, and that makes them primary producers just like plants, so that carbon can be sequestered into the soil food web by having these photosynthetic organisms fixing carbon directly in the soil. And that, of course, makes them a fantastic food source for non-photosynthetic organisms, where that carbon is available directly to organisms that either consume the algae or engulf the algae to become endosymbionts within those organisms. They also play a crucial role in rock weathering by having secondary metabolites and being able to extract nutrients, just like fungi and bacteria do via acids and other metabolic substances that allow them to gain minerals from the soil mineral matrix. And so in that case, they also play a crucial role in biochemical cycles of phosphorus, sulfur, silicon, carbon, and oxygen. They also contain phytohormones that increase plant growth and protective substances that reduce growth of pathogens, particularly root pathogens being in the soil. So they are very beneficial to plants directly. And for this reason, they are heavily studied in their role in remediating soils, anywhere from pollution detoxification or cleanup to agricultural food production in enhancing plant growth and protecting plants from diseases. They also produce hygroscopic mucilage, and that is a substance that is able to actually absorb moisture from the atmosphere, and that increases soil water retention. They also cement bind soil particles, so that means they release substances that allows for the stickiness of soil particles, and thereby they reduce and prevent erosion, and also this is another mechanism of holding water in the soil. And as a result, they reduce nitrate and other nutrient leaching and drainage, so they protect environments from runoff of nutrients into waterways and therefore causing water pollution. And they also influence soil microbial populations, their biomass activity, community composition, and diversity. That's because they are photosynthetic, so they are able to fix that carbon and interact with various microbial populations in terms of nutrient cycling, providing those extra nutrients, and as well as we have seen with, for example, protists. And the image on the right shows the biodiversity of algae in the farmlands of mid-Taiwan, and just to show you the biodiversity that can be found of these microalgae in various agricultural soils doing their job, providing all these benefits that were just mentioned. It's just a lovely image to demonstrate their presence in those soils and the amazing role that they play in the soil ecosystem. And of course, these benefits need to be elucidated and studied further so that we get a better picture of the soil food web and their role in the soil food web ecosystem, which they don't really get included in, as you would have seen by now, by the various soil food web models I have so far presented. So it's something that needs to change. Another important cycle in which soil protists play a large influence over is the silicon cycle. And this particular cycle is determined by diatoms and euglyphid testate amoebae, and that's because they contain silicon structures. They contribute to silicon cycling to the equivalent of what forest trees are capable to cycle. And the carbonate silicate cycle is part of the inorganic carbon cycle, which influences carbon dioxide sequestration via weathered mineral burial. And this you can look up for yourself because it's quite a complex process, but it actually leads to long-term transformation of silicate rocks into carbonate rocks via weathering and sedimentation. So while there's silicon cycling occurring in the soil, it is the oceans that really drive the silicon cycle. And a lot of these organisms that contain silica are present in the oceans, and their skeletons are built from silicon. And organisms belonging to the group Rhizaria, that include soil diatoms and some testate amoebae, are what really drives this silicon cycle, and it's very much related to how carbon is cycled as well. So it's very cool in a way of seeing how much of different minerals are actually sequestered to make the bodies of organisms and how then that is processed via the interaction of abiotic factors and how that relates to the geological transformation of nutrients via these biological systems. So this brings us to the end of lesson 6.9, and lesson 6.10 is our final lesson on protozoa or soil protists, and we'll continue dealing with their ecological function in the soil ecosystem. We will look at soil protist rhizosphere effects, so how they affect plants at the root level and what that means for the plant. We'll discuss how they influence plant community structure. Their role as bioindicators, so how soil protists can be studied to look at what is happening in the environment in terms of pollution and other environmental disturbances, as well as how soil protists can be used in the efforts to bioremediate pollution, what role they play in helping us to clean up polluted soils. Okay, so you'll be hearing from me again in lesson 6.10. See you then.